Good morning, students. We will continue the topic of lipid metabolism, competition number 4.2, and today's topic of discussion is de novo synthesis of fatty acid. In this, we are studying pathway, site, organs, precursor of fatty acid synthesis, enzyme complex, product which are formed. Of course, the product are fatty acid, but different types of fatty acids are formed in different tissues, and finally the regulatory steps. So first about the pathway definition. De novo synthesis means synthesis of new fatty acid. Site, it occurs in the cytoplasm. It is exactly opposite to beta oxidation, which occurs in the mitochondria. The organs in which the fatty acids are synthesized mainly are liver, adipose tissue, kidney, mammary glands and brain. Fatty acid synthesis occurs in the post-meal state of the body, and the substrate required for fatty acid synthesis is acetyl-CoA. Now, what is the source of this acetyl-CoA? The source of acetyl-CoA is glucose. In the post-meal state, glucose is absorbed from GIT. That glucose enters into the cells. In the cytoplasma, this glucose undergo glycolysis to form pyruvic acid. This pyruvate enters into the mitochondria, where with the help of enzyme pyruvate dehydrogenase complex, the pyruvate is converted into acetyl-CoA. So acetyl-CoA is formed in the mitochondria, and pathway is occurring in the cytoplasm. Before starting pathway, we should know some important features of this pathway. The first feature is the enzymes of this pathway are not free. Fatty acid synthesis requires total 8 enzymes. Out of these 8, 7 enzymes combines to form a complex called as fatty acid synthase complex. That means they are present in a complex, 7 enzymes. Only one enzyme is free. And second important thing about this pathway is the intermediates which are formed in this pathway, they are also not free. They combine with a carrier protein called as acyl carrier protein. So this is a fatty acid synthase complex. Here I am showing you 2 subunits, one above and one below. Each subunit is made up of 7 enzymes, and these enzymes are condensing enzyme, acetyl transacylase, malonyl transacylase, dehydratase, enoyl reductase, ketoacyl reductase and thioesterase. Along with these 7 enzymes, one ACP or acyl carrier protein is also present in each subunit. So each subunit has 7 enzymes, and each subunit has 3 domains. First domain is condensing enzyme, second domain is reduction domain, and third is releasing domain. In the second subunit, if you see, same 7 enzymes are present and the same 3 domains are present, but these domains are reciprocal to the first subunit. Therefore, condensing domain of one subunit, reduction and releasing domain of another subunit come close to each other. Now, this fatty acid synthase complex synthesizes 2 fatty acid at a time because it has 2 subunits. For synthesis of one fatty acid, it requires all 3 domains, but all 3 domains are not belong to same subunit. Condensing domain of one subunit, reduction and releasing domain of other subunit participate in the synthesis of one fatty acid, and in the second case, reduction and releasing subunit of reduction and releasing domain of upper subunit and condensing domain of lower subunit synthesizes another fatty acid. So two fatty acids are synthesized at a time. If we separate the two subunits, then they does not act. Therefore, dimer form of fatty acid synthase complex is only active form. Now this dimer form increases the efficiency of the pathway. That means at a time two fatty acids are synthesized on the complex. Now if we separate the two subunits, here I am showing you only one subunit. It has all 7 enzymes and all 3 domains. But this single subunit of fatty acid synthase complex cannot synthesize as fatty acid, because just now what I have told you, condensing domain of one subunit and reduction and releasing domain of another subunit synthesizes one fatty acid. So single fatty acid is inactive, or single subunit is inactive. Now first we will see how acetyl CoA is transported to the cytoplasm. Just now I have told you glucose enters into the cytoplasm, undergoes glycolysis to form pyruvate. Pyruvate enters into the mitochondria with the help of enzyme PDH or pyruvate dehydrogenase. Pyruvate is converted to acetyl CoA. So substrate acetyl CoA in post-meal state is available in mitochondria, whereas pathway occurs in cytoplasm. Therefore, we have to transport this acetyl CoA to the cytoplasm. But the inner mitochondrial membrane is impermeable for acetyl CoA. Therefore, acetyl CoA combines with oxaloacetate to form citric acid or citrate. The enzyme required for this step is citrate synthase. Now this is the first step of citric acid cycle. So when this citric acid is formed in large quantity above the saturation level, then only this citric acid crosses the inner mitochondrial membrane and enters into the cytosol. Citric acid, when it is formed in the fasting state, it is formed in low quantity. Therefore, it does not crosses the inner mitochondrial membrane. Now when citric acid enters into the cytosol, the enzyme ATP citrate lyase acts on this citric acid, and with the help of energy from ATP, it breaks the citric acid into acetyl CoA and oxaloacetate. So in this way, acetyl CoA is now available in cytoplasm. Now this oxaloacetate undergoes reduction and get converted into malate with the help of enzyme malic enzyme. And this malate crosses the inner mitochondrial membrane, enters into the mitochondria. In the mitochondria, this malate is oxidized by removal of 2 hydrogen atom to form oxaloacetate. So in this way, the transport of acetyl CoA from mitochondria to cytoplasma occurs. Once this acetyl CoA is available in the cytoplasma, then fatty acid synthesis pathway starts. So this is the step 1 of fatty acid synthesis pathway. Here malonyl CoA is formed. So acetyl CoA combines with carbon dioxide to form 3-carbon malonyl CoA with the help of enzyme acetyl CoA carboxylase. So this acetyl CoA carboxylase is the only free enzyme of this pathway. Remaining enzymes are present in complex. It is also a rate limiting enzyme of this pathway, or it is a allosteric enzyme of fatty acid synthesis pathway. Now this acetyl CoA carboxylase requires a coenzyme biotin, just like other carboxylase enzyme. Now second step is addition of malonyl and acetyl CoA to the fatty acid synthase complex. So malonyl CoA, just we have seen, which is formed, and new acetyl CoA enters into the fatty acid synthase complex. Two enzymes, acetyl transacylase and malonyl transacylase, are present in the fatty acid synthase complex. Acetyl transacylase transfer the acetyl group to the condensing enzyme after removal of coenzyme A from acetyl CoA. Similarly, malonyl transacylase also removes the coenzyme A from malonyl CoA, and only malonyl group is transferred to the acyl carrier protein. So in this way, in step 2, with the help of two enzymes, acetyl transacylase and malonyl transacylase, acetyl group and malonyl group are added to condensing enzyme and acyl carrier protein respectively. So the name of these two are acetyl CE, that is condensing enzyme, and malonyl ACP, that is acyl carrier protein. So in this way the transfer occurs. Now in the third step, condensation occurs. That means malonyl group and acetyl group combines. Before condensation, the carboxyl group of malonyl is removed in the form of CO2, carbon dioxide. So the malonyl group COCH2COOH, that COOH is removed, and now malonyl group becomes COCH2. Then the acetyl group from the condensing enzyme is added to this decarboxylated malonyl group with the help of enzyme, condensing enzyme. So in this way, the 4-carbon beta ketoacyl ACP is formed. So in this case, first decarboxylation occurs where CO2 is removed from malonyl group. Then COCH3 from condensing enzyme is transferred to that decarboxylated malonyl group to form beta ketoacyl ACP. Here at beta carbon, keto group is regenerated, therefore it is called as beta ketoacyl ACP. For this condensation, condensing enzyme plays an important role. The step 4 is reduction. Now in this step, the reduction of keto group, beta keto group, takes place by addition of 2 hydrogen atoms. When 2 hydrogen atoms are added to the keto group is converted into CHOH, that is hydroxy group. CO group is converted into CHOH by adding 2 hydrogen atoms. Now these 2 hydrogen atoms are donated by a reducing equivalent called as NADPH. This reduction requires an enzyme called as ketoacyl reductase. So in this way, the product is beta hydroxyacyl ACP is formed. Now the next step is dehydration, removal of water molecule. Now from beta hydroxyacyl ACP, 1 molecule of water is removed. So in this case, OH from beta carbon and hydrogen from alpha carbon is removed. Therefore, at beta carbon, CHOH group is converted into CH group, and at alpha carbon, CH2 group is converted into CH group. And a double bond is introduced between alpha and beta carbon. So the name of compound is alpha beta unsaturated acyl ACP. 1 water molecule is removed, and the enzyme require is dehydratase. The next step, sixth step, is reduction. Now in this reduction, again 2 hydrogen atoms are added, but this time 2 hydrogen atoms are added, one in each alpha and beta carbon atom. Therefore, the unsaturated bond present between alpha and beta carbon get saturated to form acyl ACP or butyryl ACP. As it is 4 carbon compound, it is called as butyryl ACP. The 2 hydrogen atom donor is again NADPH, and enzyme required is enoyl reductase. So in this way, after second reduction, butyryl ACP is formed. Or you can say, at the end of first cycle, 4 carbon acyl group is formed or synthesized. But body requires acyl group with more number of carbon atoms, 16 carbon, 18 carbon. Therefore, this cycle has to be repeated. That is called as cycling. During cycling, the butyryl group or butyryl ACP is formed at the end of first cycle, which is attached to acyl carrier protein. That butyryl group is transferred to the condensing enzyme. Now, when we start the pathway, the condensing enzyme binds with acetyl group. Now, at the end of first cycle, this condensing enzyme combines with butyryl butyryl group, and acyl carrier protein is free. New malonyl CoA, which is synthesized in the first step, that is synthesized by acetyl CoA carboxylase, enters into the complex and binds with the acyl carrier protein. Then the same steps get repeated. First, condensation occurs. Before condensation, again this carbon dioxide of malonyl group is removed, or carboxyl group of malonyl CoA is removed in the form of carbon dioxide. Then, so this carboxyl group is removed from malonyl group, and then the butyryl group from condensing enzyme is added to the decarboxylated malonyl CoA. So in this way, the butyryl group from condensing enzyme is transferred to the decarboxylated form of malonyl CoA. So here, now if you count the carbon length, now carbon length becomes 6. In the next step, after this condensation, the same steps like reduction, dehydration, second reduction occurs, and during which the CO group at beta carbon is converted into CH2 group, and 6 carbon acyl group is formed at the end of second cycle. So in this way the cycle goes on repeating, and at the end of 7th cycle you will get a acyl group with 16 carbon. So it is called as palmitic acid. When this palmitic acid is synthesized in adipose tissues and liver, then the last enzyme, which is called as thioesterase, will get stimulated, and it will break the bond between 16 carbon palmitic acid and acyl carrier protein and release this palmitic acid out of the complex. So in this way the fatty acid synthesis pathway takes place. Now we will see the products form. In liver and adipose tissue, the palmitic acid is formed with 16 carbon atom. But in lactating mammary glands, the de novo synthesis pathway synthesize medium chain fatty acid, that is capric acid with 10 carbon and lauric acid with 12 carbon. As these are medium chain fatty acids, they are easily absorbed from the milk and easily digested in the infants. So the product of de novo synthesis of fatty acid in liver and adipose tissue is palmitic acid, and in the mammary gland it is capric acid and lauric acid. But in some tissues, longer chain fatty acids are required. So the palmitic acid is later on elongated by two enzyme system. One is called as microsomal chain elongation system and another is called as mitochondrial chain elongation system. So first we will see about this microsomal chain elongation system, which is present in smooth endoplasmic reticulum. The enzymes of this system, elongate system, are free. That means they does not present in complex form. They are free. Second, they utilizes palmitic acid and as a substrate, and to this palmitic acid they will add malonyl CoA, and with the help of NADPH they increase the length of palmitic acid by 2 carbon to form a stearic acid. Again the same cycle is repeated. The stearic acid is taken as a substrate and malonyl CoA and NADPH are added, and again the 18 carbon stearic acid is converted into 20 carbon fatty acid. So in this way the cycle goes on repeating. The same elongation also occurs in the mitochondria. The enzyme is mitochondrial elongate system. Here again the mitochondrial elongate system utilizes palmitic acid, but instead of malonyl CoA they uses acetyl CoA. They add acetyl CoA into the palmitic acid with the help of NADH or NADPH both and ATP, and they increases the length of palmitic acid by 2 carbon atoms to form stearic acid. Again the stearic acid is elongated by adding acetyl CoA to 20 carbon, 22 carbon, 24 carbon fatty acids. So in this way the cycle occurs in mitochondria and microsome to synthesize higher fatty acids. Now this is the summary of fatty acid synthesis pathway. 1 acetyl CoA, which has initially combines with condensing enzyme, and 7 malonyl CoA of 7 cycle, 1 in each cycle, is added to the alkyl carrier protein. So it requires, each cycle requires 2 NADPH, so it requires 14 reducing equivalents, and they get converted into one molecule of palmitic acid or palmityl CoA. Palmityl CoA is the activated palmitic acid, and at every cycle 1 carbon dioxide is removed, so you will get 7 carbon dioxide molecule plus 7 coenzymes are removed, and 14 NADPH molecules are oxidized to 14 NADP molecule. Here we have seen dehydration takes place at each step, so 7 water molecules are also formed. Now regulation, first hormonal regulation and then we will see allosteric regulation. As this pathway occurs in the post-meal state, so it is stimulated by insulin, because insulin is the hormone which is present in the post-absorptive or post-meal state, whereas glucagon epinephrine in fasting state inhibit the pathway of fatty acid synthesis. Now come to allosteric regulation. Here I have told you The allosteric enzyme is acetyl CoA carboxylase. The inhibitor is palmityl CoA. What is palmityl CoA? Palmityl CoA is the end product of fatty acid synthesis, and substrate is acetyl CoA. And stimulator here is citric acid or citrate. So this is the diagrammatic representation of allosteric regulation. The allosteric enzyme, that is acetyl CoA carboxylase, is shown on the left side. It has two sites, one is active site and another is allosteric site. At the active site, substrate acetyl CoA binds and the pathway starts. But if the inhibitor palmityl CoA is present, palmityl CoA is the end product of the fatty acid synthesis. So here end product is acting as a inhibitor. Therefore it is also called as negative feedback regulation. So when this palmityl CoA is available, then this palmityl CoA binds at the allosteric site, brings the conformational change in the active site, which is shown on the right side of the figure. Due to this conformational change in the active site, acetyl CoA does not fit at the active site and the pathway stops. But if the stimulator citric acid is present, now this citric acid also binds at the other site or allosteric site, but it does not bring the conformational change in the active site. Instead of that, it increases the affinity between active site and substrate acetyl CoA, so that pathway get stimulated. So in this way the pathway is regulated. Next factor which stimulates the pathway is availability of NADPH, reducing equivalent. At last we will see comparison between beta oxidation and fatty acid synthesis site. Beta oxidation occurs in mitochondria, fatty acid synthesis occurs in cytoplasm. In beta oxidation, the intermediates binds with coenzyme A, for example fatty acyl CoA, beta keto acyl CoA, beta hydroxy acyl CoA, beta hydroxy acyl CoA. So coenzyme A is there. In fatty acid synthesis pathway, the intermediates combines with ACP, acyl carrier protein. Now enzymes of beta oxidations are independent or free, but 7 enzymes of fatty acid synthesis pathway forms a multi-enzyme complex. In beta oxidation, at every cycle the fatty acids is broken down, 2 carbon atoms splits from fatty acid and removed in the form of acetyl CoA. Whereas in the fatty acid synthesis pathway, the length of the fatty acid is increased by 2 by addition of malonyl CoA, which has 3 carbon. Why? Because 1 carbon dioxide is removed at every step. So 3 carbon atoms are added into the fatty acid synthesis complex, out of which 1 carbon is removed in the form of CO2, only 2 are added. So the fatty acid synthesis or fatty acid length is increased by 2 carbon. The coenzymes required for beta oxidation are NAD and FAD, which get reduced, whereas in fatty acid synthesis the NADPH is the coenzyme which get oxidized. So this is about the fatty acid synthesis pathway. This is in short about the pathway summary. Thank you very much.